Hello everyone, we're going to be covering for General Biology 1, Chapter 11 from Campbell Biology, and this will be covering cell communication or cellular communication. Here we're going through some of the initial images from the start of the chapter showing how any physical process that we have is actually connected all the way down to what happens to our cells. And the initial process is some signal receptor picks up usually a hormone which causes signal transduction down the line and leads to a cellular response. For our concepts for Chapter 11, Concept 1, external signals are converted to responses within the cell. This can be ancestral signaling molecules likely evolved in prokaryotes and single-celled eukaryotes and were adopted for use in their multicellular descendants down the line. Evolution of cell signaling. Research in the 1970s suggested that bacterial cells were capable of signaling to each other. Cell signaling is critical among prokaryotes. A concentration of signaling molecules allows bacteria to sense local population density in a process called quorum sensing, popular among some bacteria even in our digestive tract, being able to tell if other bacteria that are beneficial to the body are getting the resources that they need or if they need some more funneled to each other. It's amazing that the bacteria in us can even work together for our own benefit. An example of quorum sensing is the formation of biofilm. A biofilm is an aggregation of bacterial cells adhered to a surface, sort of like what happens to our teeth and gum lines. Another example of medical importance is the secretion of toxins by infectious bacteria. Interfering with the signaling pathways used in quorum sensing may be a promising approach as an alternative to antibiotic treatment. Of the major steps, first, an individual, or some examples, there is individual rod-shaped cells. In the second step, an aggregation can occur. In step 3, spore forming structures, also called the fruiting body. And then we see the fruiting bodies forming. The yeast Saccharomyces cerevisiae has two mating types, A and alpha. Cells of different mating types locate each other via secreted factors specific to each type. The binding of a mating factor to the cell surface initiates a series of steps called a signal transduction pathway. Molecular details of signal transduction in yeast and mammals happens to be very similar. For the first step, we have an exchange of mating factors between the A type and alpha type so that those cells are able to understand each other. And then the mating process involves a fusion across the cell membranes. And then we have a new A-alpha cell, a combination of both. Local and long-distance signaling. Cells in a multicellular organism communicate via signaling molecules. In local signaling, animal cells may communicate by direct contact. Animal and plant cells have cell junctions that directly connect to the cytoplasm of adjacent cells. Signaling substances in the cytosol can pass between adjacent cells. Animal cells may also communicate by direct contact between cells, surfaces, molecules, cell surface molecules. Local signaling is especially important in embryonic development, immune response, and maintaining adult stem cell populations. Here we have our plasma membrane and cell wall examples. What we see in the animal example, gap junctions between animal cells can form, whereas in cell walls there are plasmodesmata between plant cells to allow for communication. Now, for A, there are cell junctions and then, as shown above, or in B, there can be cell surface molecules that allow for the communication between two different types. It's similar to how viruses tend to invade cells as well. In other cases, animal cells communicate using secreted messenger molecules that travel only short distances. This type of local signaling in animals is called paracrine signaling. You might hear more of this in anatomy, such as endocrine and paracrine and other forms. Growth factors which stimulate nearby target cells to grow and divide are one class of such local regulators in animals. Synaptic signaling occurs in the animal nervous system when a neurotransmitter is released in response to an electric signal. Drugs used to treat depression, anxiety, and post-traumatic stress disorder, PTSD, affect this signaling process. In long-distance signaling, plants and animals use molecules called hormones. In hormonal signaling, or endocrine signaling, in animals, specialized cells release hormones, which travel to target cells via the circulatory system. The ability of a cell to respond to a signal depends on whether or not it has a receptor specific to that signal. Looking at paracrine versus endocrine signaling, think of paracrine as within the same local area, perhaps something released by one cell to be received by another cell nearby, where the endocrine signaling can involve, say, a hormone released by the brain or the adrenal glands near the kidneys, which is received further down the body, much farther than a few cells. So, local signaling, we have our target cells that are receiving information from a signaling cell, and those signaling molecules can be packed up in vesicles, secretory vesicles, that once they fuse to the outer cell membrane, they release those signal molecules, and those molecules will be called local regulators. An example of that would be an example of paracrine signaling between nearby cells. As seen in the nervous system, we have B, synaptic signaling, where an electric signal triggers release of neurotransmitter molecules. Neurotransmitter then diffuses across the synapse toward the target cell. And then for C, endocrine signaling, also called hormonal signaling, which is an example of long-distance signaling. We have signal molecules released by an endocrine cell. Hormones are then diffused into the bloodstream, and then they travel through blood vessels, eventually toward the target cell that specifically binds to that hormone, sometimes unique to that cell. The 3 stages of cell signaling, a preview. Earl W. Sutherland and colleagues discovered how the hormone epinephrine acts on cells. Sutherland's work suggested that cells received signals went through 3 processes: signal reception, signal transduction, and cellular response. In response, the target cell detects a signaling molecule that binds to a receptor protein of the cell surface. In transduction, the binding of the signaling molecule alters the receptor and initiates a signal transduction pathway. Transduction often occurs in a series of steps. In response, the transduced signal triggers a specific response to the target cell. Now, if you made it to this point in the video, likes and subscribes are you can like the video and subscribe to the channel. Neither are necessary, but they would be fun to see. And also leave a comment if you'd like, or if you're enjoying the class or learning, or would like to learn something else. Here we have an example of our signaling molecule finding the specific receptor that can actually connect to that signaling molecule, initiating signal reception. And then that combination then causes a pathway called signal transduction, where there may be multiple intermediate relay molecules that eventually lead to cellular response at the activation of cellular response. And this is just an animation that we won't be able to play unless you have your actual textbook. Or, well, your actual e-textbook. Concept 11.2, reception. A signaling molecule binds to a receptor protein, causing it to change shape. The binding between a signal molecule, called a ligand in chemistry or a ligand in biology, and a receptor is highly specific. It is very unique what kind of molecule will bind to that receptor. Or I should say, what shape of molecule. A shape change in a receptor is generally the initial transduction of the signal, basically showing that a ligand or a ligand has bound to the receptor. There's some physical change in its structure, which causes some action down the line. Most signal receptors are plasma membrane proteins, but others are located inside the cell. Receptors in the plasma membrane, G protein-coupled receptors, or GPCRs, are the largest family of cell surface receptors. Most water-soluble signal molecules bind to specific sites on receptor proteins that transmit information from the extracellular environment to inside the cell. And here we see an example figure of that plasma membrane protein that spans across the entire plasma membrane. Here we have beta-2 adrenergic receptors. A molecule that mimics a ligand can also fit into that space, which might interfere with the function of that receptor. And here we see cholesterol helping keep things together, whether or not it transfers across the membrane or stays there. There are three main types of membrane proteins. First would be G protein-coupled receptors. The next would be receptor tyrosine kinases. And then, of course, there are ion channel receptors. G protein-coupled receptors are cell surface transmembrane receptors that work with the help of a G protein. G proteins bind to the energy-rich guanine triphosphate, GTP, a similar molecule to our energy molecule, adenosine triphosphate, ADP, or ATP. G proteins are all very similar in structure. GPCR systems are extremely widespread and diverse in their functions, though. Such as examples, signaling molecule binding site, where we have multiple loops across the membrane, segment that interacts with the G protein inside the cell, shown on the bottom right, and then the G protein-coupled receptor as a whole. All those different coils, perhaps there's only one section of that exposed protein that really interacts with the G protein. But the remaining coils are necessary to anchor the cell membrane into this plasma membrane and to help give it function. Such an example, we have our G protein-coupled receptor, and then there is GTP, which binds, and then it becomes GDP, or guanine diphosphate, and then there is a G protein that is inactive, and we have our enzyme. Once the activated receptor binds to its signaling molecule, that causes the reaction of GTP, forming GDP, and then next, or I should say, we then have GTP, essentially GTP that was in the cellular fluid, transfers a phosphate to GDP in the inactive G protein, allowing that third phosphate to now be on the activated G protein. Then figure 3, that activated protein with a GTP component then binds to our enzyme to activate that. And then the energy stored in that third phosphate, as that bond breaks, and that phosphate becomes inorganic phosphate and is released back into the cellular fluid, GTP on the G protein gets converted back to GDP, and after the enzyme is activated, it then goes back to eventually being inactivated. Now the next receptor tyrosine kinases, sometimes referred to as RTKs, are membrane receptors that catalyze the transfer of phosphate groups from ATP to another protein. In biochemistry, if you ever hear the word kinase in a protein, that always involves the transfer of a phosphate group. A receptor tyrosine kinase can trigger multiple signal transduction pathways at once. Abnormal functioning of these receptor tyrosine kinases is associated with many types of cancers. Here we have an example where in the top left, a signaling molecule, the ligand, is approaching an alpha helix, or is approaching the ligand binding site. Now, the part that keeps the receptor tyrosine kinase protein anchored in the plasma membrane are going to often be those alpha helixes. And then the tyrosine components exposed inside the cytoplasm of the cell. And then these are the parts where the receptor tyrosine kinase proteins are currently in the inactive monomer state. Once the signaling molecule binds, as shown in the left side, we're going to form dimers from two different monomers of those receptor tyrosine kinases. As that dimer forms, this activates the tyrosine kinase regions, where the unphosphorylated dimer component, ATP that is in the cellular membrane, the cellular fluid, then travels toward these dimers, these unphosphorylated dimers, and transfers a phosphate group onto those exposed dimers, tyrosine components, converting the cellular ATP to ADP, 6 units of ATP to ADP. And now we have our fully activated receptor tyrosine kinase phosphorylated dimers. Phosphorylated meaning that the dimers now gained a phosphate group. And then those allow for the activated relay proteins, which usually in the cell are inactivated until finding those phosphate groups on the tyrosine, receptor tyrosine kinase dimers. You have an initial cellular response, as well as a second cellular response. Next, what we are going to talk about are ligand-gated ion channel receptors, and they act as a gate that opens and closes when the receptor changes shape. When a signal molecule binds as a ligand to the receptor, the gate allows specific ions such as sodium +1 ions or calcium 2+ ions through a channel in the receptor. An example here, on the top left, we have a signaling molecule, the ligand, which once it binds to our ligand-gated ion channel receptor, it converts it from the closed position to the open position, and through diffusion, those ions that are in higher concentration outside the cell, in this particular example, are then able to flow into the cell where there's a lower concentration of those ions, and they can allow for some cellular response down the way. And then once the signaling molecule leaves that binding site, the ion channel closes to allow no more diffusion of that specific ion. There are intracellular receptors. Intracellular receptor proteins are found in the cytoplasm or nucleus of individual target cells. Small or hydrophobic chemical messengers can readily cross the membrane and activate receptors. Examples of hydrophobic messengers are the steroid and thyroid hormones of animals. An activated hormone receptor complex can act as a transcription factor, turning on or off specific genes. Such as an example, we have aldosterone in the extracellular fluid outside the cell. This can easily transfer past the plasma membrane for being hydrophobic. And then we have our receptor protein that once it recognizes that it is bound to aldosterone, we have our hormone receptor complex, the combination of the receptor protein and our hormone. And then this can transfer over the nucleus where our DNA is housed, turning on or off certain genes, which affects the transcription of mRNA, which then, as that mRNA strand leaves the nucleus, that can affect the formation of a specific protein during translation. Transduction, or concept 11.3, transduction. Cascades of molecular interactions transmit signals from receptors to relay molecules in the cell. Cell signaling is usually a multi-step process. Multi-step pathways can greatly amplify a signal. Multi-step pathways provide more opportunities for coordination and regulation of the cellular response. Signal transduction pathways. The binding of a signaling molecule to a receptor triggers the first step of a chain of molecular interactions. The activated receptor activates another protein, which activates another, and so on, until the protein producing the response is activated. At each step, the signal is transduced into a different form, commonly a shape change in a protein. Protein phosphorylation and dephosphorylation. Phosphorylation and dephosphorylation of proteins are commonly used in cells to regulate protein activity. Protein kinases transfer phosphates from ATP to a protein, a process called phosphorylation. Phosphorylation. Many relay molecules in signal transduction pathways are in fact protein kinases, creating a phosphorylation cascade. For example, we have our receptor that has bound to its signal molecule, and that starts releasing an activated relay molecule. Once that meets our inactive protein kinase 1, that activates the protein kinase 1 protein. And then that allows for the transfer of a phosphate group from cellular ATP, forming ADP, and then that phosphate then gets bound to the inactive protein kinase 2, activating it by the addition of a phosphate group. And then eventually we have our inactive protein of interest, which, thanks to the activated protein kinase 2, a phosphate group from cellular ATP can transfer onto that inactive protein, activating it, causing a cellular response. And then we essentially have a dephosphorylation protein that removes that phosphate group, deactivating that protein, And there's also a deactivation step across all the other proteins as well to revert them all to inactive states. This was an example of phosphorylation cascade, where the activation of one protein allows for further activation of other proteins down the line through multiple series of phosphorylations. Protein phosphatases rapidly remove the phosphates from proteins, a process called dephosphorylation. This phosphorylation and dephosphorylation system acts as a molecular switch, turning activities on and off and up and down as required. For those of you who go into biochemistry, you may hear of proteins that participate in gluconeogenesis, sort of the reverse of glycolysis. Essentially, there are some situations where a protein can be both a kinase and a phosphatase. Essentially, it just depends on what side of the protein is activated, thus allowing for it to have a certain function. Now, small molecules and ions as second messengers. Many signaling pathways involve second messengers. These are small, non-protein, water-soluble molecules or ions that spread throughout a cell by diffusion. Second messengers participate in pathways initiated by GPCRs and RTKs. Cyclic adenosine monophosphate or cyclic AMP and calcium ions are common second messengers. Cyclic AMP, also called CAMP, is a small molecule produced from ATP and is one of the most widely used second messengers. Adenylyl cyclase is an enzyme in the plasma membrane that converts adenosine triphosphate to cyclic adenosine monophosphate in response to an extracellular signal. Here what we see here is ATP, and then once it crosses path with adenylyl cyclase, this forms pyrophosphatate and two bound phosphate groups, and then we have our cyclic adenosine monophosphate molecule, and then we have our other enzyme phosphodiesterase, which absorbs a water molecule to break up that cyclic bond, so that we have adenosine monophosphate or AMP. Many signal molecules trigger formation of CAMP. Other components of CAMP pathways are G proteins, G protein coupled receptors, and protein kinases. CAMP usually activates protein kinase A, which phosphorylates various other proteins. Further regulation of cell metabolism is provided by G protein systems that inhibit adenylyl cyclase. Here what we have here is our G protein coupled receptor, and the first messenger, the signaling molecule such as epinephrine, binding to that receptor. And then this allows for the transfer of phosphate from a freely floating cellular GTP onto the GDP component of the G protein, activating it, and then that can go toward adenylyl cyclase, activating that cyclase protein. Once activated, ATP in the cellular fluid, a phosphate group can transfer from that onto to form cyclic AMP, which acts as a second messenger that eventually activates protein kinase A, that eventually leads into a cellular response. Understanding the role of CAMP in G protein signaling pathways helps explain how certain microbes cause disease. The cholera bacterium Vibrio cholerae produces a toxin that modifies a G protein so that it is stuck in the active form. This protein continuously makes CAMP, causing intestinal cells to secrete large amounts of salt into the intestines. Water follows by osmosis, and an untreated person can soon die from loss of water and salt. Calcium ions and inositol triphosphate, or IP3. Calcium ions are used widely as a second messenger, even more so than CAMP. Calcium ions can function as a second messenger because its concentration in the cytosol is normally much lower than the concentration outside the cell. A small change in the number of calcium ions thus represents a relatively large percentage change in calcium concentration. Here what we have is the endoplasmic reticulum inside cells. In the plasma membrane, cellular, we have calcium pumps along the mitochondria, as well as along the endoplasmic reticulum. And ATP can affect the transfer of calcium, and ATP can also affect calcium pumps that go between the cytosol and the outer components, the extracellular fluid. Normally, the extracellular fluid is very high in calcium, while the cytosol is very low in calcium. A signal relayed by a signal transduction pathway may trigger an increase in calcium in the cytosol. Pathways leading to the release of calcium involve inositol triphosphate and diacylglycerol as additional second messengers. Diacylglycerol is also called DAG or DAG. These two are produced by cleavage of a certain kind of phospholipid in the plasma membrane. And here we have another example of our figure, the extracellular fluid, where there is a signal molecule acting as the first messenger, binding to that G protein-coupled receptor that allows for the activation of a G protein, and then the activation of phospholipase C going toward PIP2, and DAG allowing for the formation of IP3 as a second messenger, and as it travels to IP3 gated calcium channels along the endoplasmic reticulum, where there's a lot more calcium ions, calcium would travel outside the ER into the cytosol and act as a second messenger for various proteins that then get activated, leading to various cellular responses. Concept 11.4, cellular response. Cell signaling leads to regulation of transcription or cytoplasmic activities. The cell's response to an extracellular signal is called the output response. Nuclear and cytoplasmic responses. Ultimately, a signal transduction pathway leads to regulation of one or more cellular activities. The response may occur in the nucleus or in the cytoplasm. Many signaling pathways regulate the synthesis of enzymes or other proteins, usually by turning genes on or off in the nucleus. The final activated molecule in the signaling pathway may function as a transcription factor. Such as in the figure here, we have our growth factor that binds to its receptor in the plasma membrane. The initial signaling molecule here, a growth factor, triggers a phosphorylation cascade. Once that phosphorylation cascade occurs during signal transduction, once activated, the last kinase in the cascade will then enter the nucleus. Here we have inactive transcription factors that are then activated by that last kinase. And then the active transcription factor, which is phosphorylated, can then simulates transcription of a specific gene, and then resulting in mRNA then that strand of mRNA directs the synthesis of a particular protein in the cytoplasm outside the nucleus. Other pathways may regulate the activity of proteins rather than their synthesis. For example, a signal could cause opening or closing of an ion channel in the plasma membrane, or a change in the activity of a metabolic enzyme. For example, here we have signal reception, the binding of epinephrine to a G protein coupled receptor, which overall becomes one molecule. The signal transduction leads to inactive G protein becoming activated, and this can easily result in the activation of 100 molecules, which then activates inactive adenylyl cyclase. And then active adenylyl cyclase leads to the formation of cyclic AMP from ATP. This can be propagated 10 to the fourth power, or 10,000, which then leads to the activation of inactive protein kinase A, which then leads to inactive phosphorylase kinase becoming activated in about 10 to the fifth amount, or let's see, 10 squared is 100, 10 to the third is 1,000, 10 to the fourth is 10,000, 10 to the fifth would be 100,000 activated phosphorylase kinases. And then this leads to 10 to the sixth, or 1 million activated glycogen phosphorylase molecules. And then what we have is the cellular response, where glucose 1-phosphate about 10 to the 8 molecules, or 100 million molecules, then form glycogen. Amazing that just one single binding add during cellular reception can have such a profound impact of activating such a wide amount of other molecules down the line. Signal receptors, relay molecules, and second messengers participate in a variety of pathways leading to nuclear and cytoplasmic responses, including cell division. Regulation of the response. A response to a signal may not be simply on or off, therefore aspects of signal regulation, such as amplification of the signal, and in turn the response, specificity of the response, overall efficiency of response enhanced by scaffolding proteins, and fourth, termination of the signal. In signal amplification, enzyme cascades amplify the cell's response to the signal. At each step, the number of activated products can be much greater than the preceding step. The specificity of cell signaling and coordination of the response. Different kinds of cells have different collections of proteins. These different proteins allow cells to detect and respond to different signals. The same signal can have different effects in cells with different proteins and pathways. Pathway branching and crosstalk further help the cell coordinate incoming signals. Here in our example figure, we have our signaling molecule binding to our plasma membrane receptor, which then leads to relay molecules being transferred. And then we have our first response. Cell A pathway leads to a signal to a single response. Now, in a different example, a similar pathway leads to response 2 and response 3. Cell B, the pathway branches leading to 2 responses. And cell C, we have 2 different receptors with 2 different signals that each release a relay molecule, which then come together for the activation or inhibition of something else. Cell C, or I should say, cell C has crosstalk occurring between 2 pathways, and perhaps one activates and one inhibits. And then in response 5, we have cell D, where a different receptor leads to a different response. Signaling efficiency, scaffolding proteins, and signaling complexes. Scaffolding proteins are large relay proteins to which several other relay proteins are attached. Scaffolding proteins can increase the signal transduction efficiency by grouping together different proteins involved in the same pathway, so that as one component, one intermediate relay molecule is released from one, that can be uptaken by another protein more quickly. In some cases, scaffolding proteins may also help activate some of the relay proteins, such as our signaling molecule binding to receptor. And then we have our scaffolding protein, which helps bring 3 different protein kinases toward the receptor and keep them all grouped together for their functions to be used in tandem. Termination of the signal. Inactivation mechanisms are an essential aspect of cell signaling. If the concentration of external signaling molecules falls, fewer receptors will be bound. Unbound receptors revert to an inactive state. And that helps to save on cellular resources so that a cell isn't constantly just producing something when it's no longer necessary. Now we have concept 11.5. Apoptosis requires integration of multiple cell signaling pathways. Cells that are infected, damaged, or at the end of their functional lives often undergo programmed cell death. Apoptosis is the best understood type. Components of the cell are chopped up and packaged into vesicles that are digested by scavenger cells. Apoptosis prevents enzymes from leaking out of a dying cell and damaging neighboring cells. Apoptosis in the soil worm Caenorhabditis elegans. In worms and other organisms, apoptosis is triggered by signals that activate a cascade of suicide proteins in the cells destined to die. In C. elegans, a protein called CED9 or SED9 is in the outer mitochondrial membrane, serves as a master regulator of apoptosis. SED9 acts as a brake in the absence of a signal promoting apoptosis. When the death signal is received, an apoptosis inhibiting protein, like SED9, is inactivated, which disables the brake. The apoptotic pathway then activates proteases and nucleases, things that cut up proteins and DNA of the cell, respectively. The chief caspase in the nematode is called SED3. Here we have in figure A on the left, a receptor for death signaling molecule is not activated, so there's no death signal. And we have our active SED9 protein, which inhibits SED4 activity, SED9 on mitochondria. And then inside the cell, there are SED4 and SED3 inactive proteins. Once the death signal binds to that initial molecule, inactive SED9 no longer inhibits SED4. Active SED4 and active SED3 then lead to an activation cascade, activating nucleases that chop up DNA and proteases that chop up proteins. And the cell then forms blebs during the cell death signal. Apoptotic pathways and the signals that trigger them. In humans and other mammals, several different pathways involving about 15 cascades can carry out apoptosis. Apoptosis can be triggered by signals from outside the cell or inside it. Internal signals can result from irreparable DNA damage or excessive protein misfolding. Apoptosis evolved early in animal evolution and is essential for the development and maintenance of all animals. For example, apoptosis is a normal part of development in hands and feet in humans and paws in other mammals. Apoptosis may be involved in some diseases, like perhaps Parkinson's and Alzheimer's. Interference with apoptosis may contribute to some cancers. Here we have interdigital tissue, cells undergoing apoptosis, allowing for those regions where cells are multiplying to branch out further, while the apoptotic regions are staying separated, and this allows for the space between digits to form, between hands and toes. And then we're back to our example of a gazelle or a deer, an animal running from a cheetah. And here I believe we might have just a bunch of our figures from the chapter. Lay's chips, potato chips, and Dorito chips, perhaps representing activation or different signaling molecules. In any case, that concludes Chapter 11 from Campbell Biology for General Biology 1. And if you definitely made it to this part of the video, please leave a comment on the value you got from the video, anything you would like to see in a later chapter. And if you have the interest, like the video and subscribe to the channel to help it grow from where it is right now in its sub 200 subscriber level. In any case, as always, wishing you all the best in all your classes, wherever they are, and peace.